All right, let's talk about some more pathways here. Um, some other or another fate that glucose can undergo for metabolism is called the pentose phosphate pathway. Now, our book, I'll be completely honest with you, our book doesn't go into a lot of detail about the pentose phosphate pathway. She just kind of mentions a couple of things, and I've really condensed it here into these two slides. So I've got two slides. It says here the pentose phosphate pathway is an alternative to glycolysis for glucose oxidation. So it's just another way that glucose can be metabolized. Now, you might be wondering, why would I need another pathway? Well, there's a good reason for this. Okay, the reason why you need to have a second pathway is because of the intermediates that are produced along the way. Okay, when we do glycolysis, we have some intermediates. We looked at all those intermediates, but if we need NADPH, for example, which is a coenzyme that's used in biosynthesis of things like cholesterol and fatty acids, well, if we need that molecule, that's going to be produced in the pentose phosphate pathway. Also, in the second stage of the pentose phosphate pathway, which again is just another pathway for the catabolism of glucose, we end up producing this molecule here, ribulose 5-phosphate. And if you look at the third step, you produce a molecule that I think is even more interesting, ribose 5-phosphate, which is used for the synthesis of nucleotides like ATP. There we go. As it says here, ribose 5-phosphate sugar is critical for nucleotide and nucleic acid synthesis. So really, all you need to know about the pentose phosphate pathway is that it's an alternate pathway for the catabolism of glucose, and that it provides intermediates. The only one that I would ask you to have memorized is NADPH, because it's going to come up a lot later on in the class. It'll provide intermediates like NADPH and other intermediates that are useful or required in biosynthesis. That is literally all you need to know about the pentose phosphate pathway. Here are the stages, or the major stages of the PPP, the pentose phosphate pathway. Again, we start out with glucose. It gets converted into G6P. And in the first stage, we end up producing NADPH, which I told you is an important coenzyme in some biosynthesis, okay, of things like fatty acids and cholesterol. That's why this occurs in the liver, right? The liver is where cholesterol synthesis occurs. And then I told you that this intermediate here, ribose 5-phosphate, is used in nucleotide synthesis. All right, that's about it for the pentose phosphate pathway. All right, let's talk about something different, gluconeogenesis. So students sometimes get, let's think, we got glycolysis, gluconeogenesis, glycogenesis, and glycogenolysis, okay? You need to know the difference between all those. And if you're like, what were those again? Look, we've got glycolysis, okay? That's what we've talked about tonight, mostly. The reverse of glycolysis, okay, is gluconeogenesis. Gluconeogenesis. Okay, those are opposites, right? Glycolysis is breaking down glucose. Gluconeogenesis, as you can see from the title of this slide, is synthesizing glucose. So we're going to be making glucose, okay, from non-carbohydrate starting materials. The other two that I mentioned were glycogenesis and glycogenolysis. That's something completely different. It's got nothing to do with this section. I'll write them down here. I'm going to use a different color. Glycogenesis, and glycogenolysis. Okay, those are also opposites of each other, but those have to do with the molecule glycogen. So glycogen is what they are dealing with. And glycolysis and gluconeogenesis, those have to do with glucose, right? So that's what they have to do with glucose. Now, these two down here, which again, we're not really worried about at the moment, but glycogenesis is going to be the synthesis of glycogen, and glycogenolysis will be the degradation of glycogen. The reason I bring those up because it's four terms that all kind of sound similar. And if you're the, if you find them a little bit confusing, you wouldn't be the first student to find it. Okay, that would be crazy to say, oh yeah, you're the only person who's ever thought that. No, not at all. I thought that for a long time. Okay, so look, under certain conditions, like starvation or following strenuous exercise, you've consumed all your glucose. You've ever heard of hypoglycemia? This is when we have low blood sugar. We're going to need to either make glucose or replenish it through our diet. Well, if you can't consume sugar fast enough, or you can't get any sugar, well, you're going to have to make it. All right. So in that case, you need to rely on this process called gluconeogenesis, right? I told you that the human brain requires something like 20 grams of glucose per hour. Okay, so that's a lot of glucose. So gluconeogenesis, again, makes glucose from non-carbohydrate starting materials like lactate, conjugate base of lactic acid. I mean, we just looked at the structure of lactate. Glycerol, everybody here should know the structure of glycerol and have that well memorized. Most amino acids, not leucine and lysine, the exceptions aren't important. Glycerol and amino acids are only used in starvation conditions. So these last two, I mean, if you're really hard up, if you're starving, you're going to take glycerol and amino acids and you'll convert them into glucose. And that's to keep yourself alive, right? If your brain isn't functioning, you can't survive. Okay, and this process occurs primarily in the liver. Now, something I should draw your attention to before I move on is that I told you that glycolysis and gluconeogenesis were the reverse of each other. It's not true. That's a lie, actually. Okay. In fact, I think it's one of the questions on your practice exam. It's something like, which statement is false? And one of the statements says, gluconeogenesis is the reverse process of glycolysis. That is false. Right? Glycolysis is a 10-step pathway. There are several steps if you compare glycolysis and gluconeogenesis, there are several steps that are the reverse of one another, but there are some steps that are not. And so the pathways are not exactly opposite. Gluconeogenesis is one of several main mechanisms used by humans and other animals in order to maintain our blood sugar levels to avoid hypoglycemia. This is a simplified version of gluconeogenesis. I just ripped this off the internet somewhere. I thought it was pertinent to what we're looking at. We can take things like fatty acids or amino acids, or more probably lactate if we're not starving. We can convert that lactate into what? Pyruvate. And then we can put that pyruvate in glycolysis backwards, right? We're kind of doing the reverse of glycolysis here. And again, I told you it's not exactly the reverse, but it's close. You make G6P, you cleave off the phosphate, and you end up with glucose. All right. Well, again, I took this off the internet. Not everything on the internet is true. It's not exactly the reverse of glycolysis. We'll have to look at it in a little more detail. Okay? Here's a summary. If you compare the two processes, it says they're basically the opposite. Okay? But basically doesn't mean they're the exact opposite, does it? Basically, the opposite processes. Glycolysis, but glycolysis and gluconeogenesis are not just a simple reversal. What you need to remember is steps 1, 3, and 10. Okay? The three non-reversible steps of glycolysis must be bypassed by new routes. Okay? So these are the steps that we're concerned with. Steps 1, 3, and 10. Okay? If you look at gluconeogenesis, so starting with pyruvate, I mean, if you look at this step here, let's say this step here, we go from 2-phosphoglycerate to 3-phosphoglycerate. I mean, that's the exact reverse, right? If we look at this step here, if we go from 3-phosphoglycerate to 1,3-bisphosphoglycerate, that's also the exact reverse. But what I want to point out is that steps 1, 3, and 10 are not the same. Right? When we go from glucose to G6P, we use hexokinase. Here we have a different enzyme. When we go from G6P to glucose, we use this enzyme glucose 6-phosphatase. Something completely different. Right? Here we use fructokinase. Now we get something completely different. And if you look at the 10th step, wow, it's really all over the place. We even have an intermediate we didn't even see in glycolysis. Oxaloacetate wasn't even present in glycolysis. Those steps 1, 3, and 10, are irreversible steps. Okay. So, step 3 of glycolysis was catalyzed by PFK, phosphofructokinase. What's it going to be stimulated by? High amounts of adenosine monophosphate, diphosphate, inorganic phosphate, right? Because that's telling the body we got low energy. What's it going to be inhibited by? High amounts of ATP, right? If we've got a lot of ATP, why would we need step 3 of glycolysis to occur? We don't. Okay. Well, what about in gluconeogenesis? Well, not only do we have a different enzyme, it's not PFK, it's this enzyme fructose 1,6-bisphosphatase. It's going to be the opposite. Instead of being inhibited by high amounts of ATP, that enzyme is going to be stimulated by high amounts of ATP, right? Because at times of excess energy, gluconeogenesis is going to be favored, right? If we have a lot of ATP, if we've got too much ATP, why would we need to be metabolizing glucose? We'll just make it, right? And then we'll store it. The Cori cycle, I don't think I've ever asked any questions directly about the Cori cycle, maybe like a very simple question. But if you think about the lactate in skeletal muscle, from anaerobic metabolism, okay, so from fermentation, that lactate is going to get transferred into the liver, okay? So let's follow it here. So the lactate, where am I here? Okay. Lactate is going to get transferred into the liver, okay, in our gluconeogenesis, okay, and gluconeogenesis is going to occur in the liver. So our lactate is going to get converted into pyruvate, finally G6P, right after all the steps, okay, so this is all the steps of gluconeogenesis. Then it's going to be acted upon by the phosphatase to produce glucose, which can go back into our muscles and be converted back into pyruvate, and we can harvest ATP, or we can store it as glycogen. So this cycle of glucose in between the muscles and the liver is called the Cori cycle. Here's my summary of the Cori cycle. I know there's a lot of words on here, but I find it's just the best way to help my students kind of summarize what the heck the Cori cycle is. So look, let's just go over it together quickly. It says the metabolic pathway, which lactate is produced by anaerobic glycolysis in the muscles, moves to the liver. So we take that lactate, get it back to the liver, gets converted back to glucose by gluconeogenesis. Then it can go back to the muscles. Okay. Well, once it's back to the muscles, it's going to get broken down, it's going to undergo catabolism to produce ATP, and it can be converted into more lactate, or it can be used to replenish muscle stores with glycogen, which is a polymer of glucose. Overall, the glycolysis part of the cycle produces 2 ATP molecules at a cost of 6 ATP molecules consumed in gluconeogenesis. This is something I neglected to say, that gluconeogenesis costs a lot more energy than you make in glycolysis. So it's not a sustainable process. So each iteration of the cycle has to be maintained by a net consumption of 4 ATPs. Therefore, the Cori cycle cannot be sustained indefinitely. This is just a little cartoon of the Cori cycle, which shows you how glycolysis produces 2 ATPs, but when we do gluconeogenesis, it costs us a total of 6 ATPs.